All right, today we are going to cover several different topics. So those topics are a little bit diverse. They are similar, unrelated, but each covers a slightly different perspective, especially the last one. If you have never heard about the single-cell immunoraptor analysis, anyone have ever used that before? Single-cell immune cell raptor profiling? No idea at all. Okay, so this is something we will cover, and the first one will be the batch effects correction. Another is one of the methods people use for batch effects correction, but it's more generalized approach for you to align two different domains of the information. It's called canonical correlation analysis. So I hope you can get some idea about the principle of this analysis. Just like you learn PCA, you know it's rotation of the matrix. But now this is two matrices and rotate at the same time. Then we will discuss trajectory analysis and see how can you build pseudotime if you believe your cells under study are at a different stage of development, or after perturbation, some cells start to respond, some have a lagging and behind, so you create a trajectory among cell states population. So the batch effects people used widely in single-cell data study. This is a publication in Nature Biotechnology. As example, they have control cells, and after the control cells, they use this IFN beta to stimulate the cells, then measure the perturbed cell population again. That's what you normally will see, the cells separated into two populations, color-coded indifferently. What they try to do is to apply this batch effects correction algorithm and to move those cells and their embedding position, so allow those cell populations to superimpose onto each other. They overlap more than unaligned data set. So this idea was rooted from a traditional microarray analysis for the gene expression from RNA-seq, which is not a single-cell RNA-seq. People believe there are technical bias in terms of, for example, if you use microarray, you know they are reading the plate, and if you put your sample into two different plates, and there's systemic shift of the color reading. All right, one plate may have 1.5 higher intensity for right channel compared to another plate. In RNA sequencing analysis, people believe there are batch effects because you load your sample in different time for the sequencing, and maybe prepare the RNA extraction differently, so you get a shift of bias in terms of different batches. Even though you may run the same analysis, same experiment on the same population of cell twice, you still see this shift. In that case, you believe their technology, this batch effects caused by the technical factors. So you want to bring down the two batches into the same level and then make them comparable. If I draw an illustration, see, this is a RNA-seq, for example, experiment, not single cell. So you have just one sample, so sample one. And then you have sample two, okay? And those are the genes. Gene 1, gene 2, gene N. So for the same gene, the assumption for some genes express high, some low. Okay, so this bar indicate expression because this is a conventional RNA-seq. Each sample is only one sample here. It's not many cells. So the bar indicate how high this gene expressed in that bulk RNA-seq sample. And here you expect the global mean are the same. If you put a mean here, and for this particular sample, it should be comparable with this one. Okay, so you expect globally these two means should be the same across all the profiling of all the genes. This is a big assumption because if this is the same sample, biological replicates 1 and 2, this makes sense. But if this sample 2 is a perturbation, is a mutation, for example, or you have a diseased sample, this is healthy control. If you assume the global expression are the same, this is a bigger claim. Then in that case, if you see a shift, so this one, the average shift to say 1.5, this is just 1. So globally, this sample, because of technique batch effects, you have an overall global shift of this gene expression. Every gene seems to shift a little bit. In that case, you want to bring this down. So you divide this biofactor, divide by 1.5 for all the genes expression. Then you bring the average down. So every one getting down a little bit. So make them comparable. Then you compare. So after you do this shifting, so you divide this factor. Then you make the global comparable, and then you can start to compare, say, gene 2, this compared to gene 2 here. Okay, in this case, this is downregulated. So that's the idea about it. There's an issue, however. For example, the global shift is true. You have a mutation in this and make a cell smaller or bigger. So your overall expression is supposed to be above, every gene should a little bit higher than that or lower than that. Another example is that if you collect a sample in 25 degrees C, which here is 37 degrees C. So every gene in this condition, because activity is higher in high temperature, so you expect biologically you have this 1.5 global shift. That's a global truth, and that's the ground truth. But if you apply batch effect correction, you bring it down to the same level, comparable. And you will still see some genes upregulated, some downregulated, but in reality, all the genes are upregulated. All right, so you get the idea? Nobody talk about this because we just assume in conventional RNA-seq, these two samples are comparable. Say 95% of genes, the average should be similar. You only want to find some of this. After batch effect correction, you want to find this extreme difference. So maybe a couple of 20 or 150 genes either upregulated, downregulated. I think that's fine. And you can also sometimes use housekeeping genes. So you see, okay, this is a housekeeping gene here. You will find the same housekeeping gene in this sample. Make them comparable rather than all the genes average. Again, the assumption is this housekeeping genes in two populations, two samples, have a comparable or very similar expression on average. But any condition, if this assumption is not hold, and then batch effect correction is not right, you shouldn't apply that. Okay, in single cell, there's no guarantee unless you know you run the same population of cells And it's just because of the batch effects, you know exactly that's from the technical bias. You see this shift. But even in this case, I'm not suggest you to do this because you artificially put the cell together. I don't know the purpose. Again, for single cell data, everybody just follow the routine. See, that paper people used batch effects correction, so I should apply. If I miss that, maybe I missed something important. But in reality, you introduce the strong bias. But there's no meaning you want to cheat yourself to artificially put those cells together. This is just a projection on the low dimension space. If they're separated, they should be separated. All right, so this is a comparison for several batch effects correction method. And combat, if you use conventional bulk RNA-seq, this is a method people used for RNA-seq sample batch effects correction. And the cell rate has its own, and this is called a mutual neural network, nearest neighbor network. Dana Peer, she's not a random person. She's a very important person in data analysis in single cell. She studied cancer single cell, and this is her strong opinion, and I believe it's true. So what she said is batch correction is worse for cancer single cell study because each sample is unique. I think what she's talking about is not the same sample you sequence twice. She's talking about the samples from same breast cancer patients and maybe from different location of those this patient biopsy, or maybe same cancer type but different animal model. You cannot assume those cancer cells should be after projection on the t-SNE should be overlapped onto each other. It's terrible, but if you follow routine, reviewer will say, okay, you didn't do the batch corrections. But these people should be educated. By the way, she just said cancer, but I think this rule applies to most of single cell data, okay? Again, Compact is a commonly used package, people for bulk RNA-seq, but you can just borrow that, just assume single cell is also same as the bulk RNA-seq. You can directly apply Compact, the algorithm, but mostly people use those ones newly developed for single cell data. This is the idea and behind this called the mutual nearest neighbor batch correction. Okay, so you have one side of the data, okay, X, Y, Z, and 3 different cell types, different colors. Then you have another batch get data from this, and then they shared only 2 cell types. So this sample has yellow cell type, but it's missing in this sample. So in order to correct the batch effects, you found the nearest, so after embedding, okay, low dimension of the cells. Then you look at the distance between them. And then you found the nearest distance from those cells are those group. And then you do the other way around. All the nearest neighbor for this group are there. So if they're nearest neighbor to each other, then you make a link between them. After that, you can start to merge, okay? Merge those similar cell populations, and then project the other cell types to the same space. Okay, that's just find the nearest neighbor, and also they have to be mutual nearest neighbors. So they're nearest neighbor to each other, then you align cells, put them together. So another approach is used, so this is from a UK group published in 2018, and similarly in the same journal, side by side, there's another group. They use CCA, okay, canonical correction analysis, to correct the batch effects. And these two papers described very different algorithms, but similarly, over the years, people just randomly select, I like to use this, I like to use it. It doesn't matter. But at the end, is a problem. This problem of batch effects correction maybe doesn't matter. Okay, so whatever method you want to use, you just put cells together, and it looks like they are overlapped, but in reality, you didn't change the expression matrix at all. You just changed their embedding. I still don't understand why you are doing it. We have to do that, but because for the completion of this course, I have to mention this. So you don't see later say, I have never been taught about the batch effects correction, so that's why I didn't use it. But I mentioned, I still don't know why you are doing this, why we need to do this for single cell data. Okay, the idea is here, you have two samples, then you project them on the space, you see, okay, they separated this batch and that batch. So you apply some technique here to make them aligned. One of the reasons I think is justified is after this embedding, you just call cell type together. So you say, okay, this cluster, what the cell type are, then I know the same cell type. If you don't do this, you don't know how to, but you still can annotate the cell type separately. So I still don't know why you are doing this. Just make you feel better, maybe. Okay, so this is the idea. You have two different batches, different perturbation, then you try to align them. So the technique this method uses called CCA is to correcting linear relationship between two multi-dimensional variable. So the data represented in the space matrix format. So the idea is you have this. This is more generalized. So you have data from same object, maybe different views. Okay. You have view 1 generate data view 2, and they are unrelated. It could be defined by different parameter, different types of parameter even. You'll still be able to apply this technique to project this pink, okay, data into the space, and this blue data into the space. Try to then, the semantic space, try to make them align to each other for the data points. In single cell data, this is from one condition, this from another condition, they all same set of genes. And you can also apply to, for example, this will be single cell RNA-seq, the gene expression, this will be ATAC-seq, okay? ATAC-seq just measure the open chromatin structures at single cell level as well. So you can do this alignment, although they measure different things for the different side of the cells. And example is like the different views. You have a picture of person, then you have this sound track signal from the same person. See, if you can apply this technique for the two different views, And for the picture, you have a database, then you have their corresponding soundtrack record. If they're shuffled, you don't know whose picture and whose voice. And using CCA, you probably can find this match because the image occupied this view one space, and there's some structure, and then its sound will have a corresponding structure. If this structure can be learned and projected on the same space, you will find the corresponding pairs of those samples. Another example is you have pictures. So this is one semantical view, and then you have a description of the text. So you can vectorize those pictures and also vectorize the large language model. Model is the vectorization of the data is compressed into vectors. And those vectors can be treated as a matrix, okay? Can be treated as a data points. You can project those data onto the same space to make a link between those pairs. And in the EEG and also functional MRI generate different kinds of signals, but on the same brain samples, then you can do this CCA to project those data onto the same space, then look at the corresponding relationship between data. So that's what the CCA is doing. So the idea is very similar to PCA. Okay, in PCA, we deal with just one batch of data. You just rotate yourself to display the variability among data points. But here we have two batches of data, okay? Red and green. You rotate them both at the same time. So you apply a rotation matrix to red cells, red data points. You apply another rotation matrix to green cell, green data points. At the end, you want to achieve is not to display their variabilities, try to make their correlation of the projected position as much as possible. The correlation will be computed by using the information of this X bar, Y bar, okay? So X bar is the data, X, and transpose X. So this is a covariance matrix. And you do covariance matrix from data set X and covariance matrix for data set Y. If you remember, for PCA, we also do the covariance matrix. So after you do the covariance matrix, you take eigen decomposition to change, to find the rotation matrix. Rotation matrix is composed by the eigenvectors. So in PCA, you have just one X data, okay? So you do this X transpose times X. So this is a covariance matrix. Let's call it A, okay? Then the next, you do the decomposition. So A times V, V is the eigenvector. Lambda V. So that's the form, right? And then these are the PCs in eigenvectors. So CCA doing the similar things here. but it deals with two matrices, X and Y. Let me write them upside down. So you put X and Y. You do the same thing for X. So you do the transpose X. So this becomes A. Do the same thing for the Y. So you transpose Y. Let's call it B. So here, rather than one matrix, you need information from both matrices because you want to align them. You want to look at their own variability and covariance among the same data, and also you want to know the covariance between X and Y. So there's something like X transpose times Y, Y transpose times X. So some information will be in there in order to align them. But it's still slightly different, I believe, if I transpose A. So at the end, just take a little bit of slightly different formula. So this part is the same, and the information from the X matrix, and then you have a B matrix from the Y matrix, B covariance matrix. So that's a CCA formula in order to compute those rotation matrix. There's some theory behind those, how the derivation derive those formula. Basically, you want to have a rotation matrix small a in order to transpose, to rotate your data points in X matrix, and then use B, another rotation matrix, to rotate your data point in matrix Y. And then after rotation, after projection, you want to compute the correlation between these rotated data points. And you want to make the best guess or optimization of your A and B so that the correlation value as large as possible. Basically, you maximize the correlation between this transposed or rotated data points projection. So we know the goal is to achieve the optimization of A and B values, to find them. So the correlation between these can be expressed in this way. Then this is the algorithm, and you have covariance matrix between X, Y. So that's between these two. And also covariance matrix of the X, Y themselves. So that's your input, okay? This can be easily computed. Then you apply this transformation. This is use SVD decomposition. Eigendecomposition is similar, okay? Then you return the matrix, the projected after rotation, okay? You get A and B. So in MATLAB, you can put these two matrices in, and A and B are the return, okay? And those are the rotation matrix. A and B are the so all these algorithms are related, like PCA, okay? If you consider there's a B, assume there's a B here. And then if this B becomes an identity matrix, then you can just cross them, and that's the formula for PCA. That's why in this table, the B for PCA is identity matrix, just no information from another matrix. You only use covariance matrix from A, from X, okay? Then CCA, you use information from both X and Y and XY, okay? So this is the B. There are several other algorithms that use very similar framework. It's all about the eigenvalue equation. So A multiplied by eigenvector, and that eigenvector, this product equal to the eigenvalue times this eigenvector. So this is eigendecomposition formula. So this equation and help you to find the solution of your rotation matrix to give the distance after your rotation that between these two are the smallest. So correlation is also largest. So the same idea. So you express slightly differently and translate that into an optimization problem. Okay. So there are also different modification of the conventional CCA. You can apply the kernel, and if you believe the variables in those X matrix, there are some nonlinear relationship. There are not. So you apply kernel function to transform, basically transform your input data. And there are special versions to deal with the data contains a lot of zeros. Okay, there's sparse. There are other different CCAs. So this is a formula of CCA, and you try to maximize linear. This is a conventional CCA, and then this is after you apply kernel function, then do the same thing. In ecology, there's another analysis also called CCA, but it's very different from what we talk about here, canonical correlation analysis. That CCA is called canonical correspondence analysis. Okay, just make sure they're not the same. Another technology we can apply if you have good examples. Just be aware of this there, especially if you have time series data. This is called dynamic time warping. In this case, you have two time series signals. They are sampled in different frequency. So one and another is supposed to be from the similar signals, but they have to be aligned. Then this method will take these two vectors of signal and try to match them, so you can have match the signal in terms of frequency and the shape. The example will be illustrated very well with this one. You have one sample from this blue curve. Okay, and then you have another signal, okay, from this orange curve. After you put these two vectors into this function, okay, dynamic time warping, you will get the minimized distance between these two vectors after you align them. And the distance becomes really small. If you directly compute the distance between them, it's huge. But this, after you align them, then you have output, this aligned X, aligned Y, And if you plot them out, you look at something like this. So just imagine if you have data, time series data, we will mention about maybe later this pseudo time series data. And there's technology you can use to align your pseudo time or time series points from two different data sets. Okay. So inside this is the algorithm take shortest distance. So this is how the align was did. Okay, we move to another topic about the pseudo-time analysis. So biologists interested in to track the individual cell development. If we have a camera, if we can take continuous sample from individual cells, it will be ideal, and we know how the gene expression program change in the early stage, in the middle, and after later stage. And we can study the development to study homeostatic tissue turnover, okay, stem cell function, etc., or cancer development. So people develop those image-based or sequencing-based technologies to track individual cells' trajectory in vitro and in vivo systems. So you can have markers, you can have mutation points, whatever, to track those differences. However, single cell is a snapshot of one time. There's no continuous sampling because you disrupt the cells through the dissociation and the barcoding and the cells lysis. There's no way you can continuously read out mRNA abundancy from the healthy individual cells. So people use this idea, say, okay, when you sampled hundreds, thousands of cells, those cells by themselves are not in the same development stage. Some cells have been there long enough, become older, and some younger. So if you can look at their gene expression profile and align them based on the assumption of the continuous development, there's a timeline between them. You just assume you know this timeline, and it's a continuous steps that you can align cells, follow this timeline to put them so you can compare the cells between neighbors or across different timeline points. So we know the biology system is very robust, and this called this Waddington's idea is called what is called canalization, right? So you have the early stage of development of cells, and you can differentiate them into different cell types, but they are not going randomly cover all the landscape of the development landscape. They will have some of those canals, and you go either this particular cell type or that particular cell type. And even you have a genetic perturbation or environment factors. Those trajectories very stable. Okay, you want to derive to that direction, you will not jump after a certain breaking point. You will not jump from B cell to T cell development stage. They will go to these trajectories. So because of those canalization effects existing, so we can assume the population cell you obtained just follow a couple of the trajectories, not everywhere, not a network. Okay? So this is the idea, try to infer those trajectories to see whether some of them maybe not true, but hopefully some of this trajectory will reflect real biological process. Okay, any of those biological process can be recovered will be very useful to understand the cell population differentiation development. So this is an illustration. You don't have physical time. You don't have those. You can sample at different time points, but you just cannot sample the same cell. Or, yeah. So if you have some real time points, will be very helpful to help you to do this. But in reality, we have mostly this single cell data which lost the temporal information, but you want to construct this pseudo time. And after you have this, you can follow individual genes change from one cell to another, but they are not randomly shuffled. They follow this trajectory. So you hope some of the genes will be upregulated across the time, and some will be downregulated. So those genes' response or correlated with pseudo time will be valuable genes to understand the procedures of this development of different stage of the cell. Is that clear? No questions, right? There's some good data set which we do have time. We collected, for example, the human embryonic stem cells from time 0, time 4, time 6, until it's developed into this, what's the EPC? So endothelium cell, some kind of so you can follow this to construct cell population contains some of the real data time points. That will help you to build those trajectory. And in reality, it's still very hard because the construction of the projection of t-SNE will not have a very clear trajectory. So some other technique maybe we can use. So this is the ideal situation. You found those lineages, and from here to here, from here you can infer those difference. And I think those are the different differentiation protocols, and create similar trajectories that give you the confirmation that this procedure is strongly canalized, and you can reconstruct the real trajectory. So if you compare different differentiation protocols, and this paper found, okay, using this system, and you have through this trajectory, and using this 5-factor differentiation protocols, and those marker genes changed and differently from this pathway, but at the end they achieve similar results. They all derive those endothelium cells, but the different protocols derive cell through different pathways. That's what we can learn from this paper. Yeah, this is another illustration how the single cell trajectory analysis will help you. So in order to take this picture, you have to either follow at different time point to take picture and Photoshop, put them together. But in our single cell data, you probably take a picture and the cells are in different development stage. So if you can align them, you just reconstruct this by one picture, okay? Same idea, and if you don't have a video recorder, you cannot take this series of pictures in order to study this horse gait. But if you have a picture for many different running horses, and then you just align them and to reconstruct from that population picture of the horse running, if they're running in the same way, right? You can have this trajectory aligned. Same idea, we need to convert t-SNE plot and into something like this, okay? Not just clusters. Clusters will not tell you the relationship between cells. So we need some data manipulation and use different ways to project data to emphasize the similarity and also the trajectory, the change of the one similarity to a different level of similarity to increase this resolution. One of the idea is after you use PCA or t-SNE plot, you start to build these cell networks, cell center networks. Okay, you think there's one class of cells in green, so that the center they're stem cells, and then move away from stem cell, they're T cells, early stage. Then you put a focal point there. See, there's subpopulation of cells, so you make a link. At the end, you create these networks. Then you just follow the network, see, okay, I can move from this node to here, to here, to here. You just artificially create this parse through this network. Okay? So that's the pseudo-time trajectory you created. And hopefully, you can find cells in the different bins, and you label those bins with the time, okay, based on where they're located. You project them onto this parse, and then just bin them, say, okay, cells, even in the same bin here, there are some early, some late. You can just put a number on it. So this is the same idea, but use a UMAP. You try to find. This is a network base. Okay, so those are the basic idea of the pseudo-time construction. So it's based on the dimensionality reduction first. So you can use PCA, you can use t-SNE, okay, all the technology you can use. And then you start to construct a network. And then after the network, you run an algorithm called the minimum span tree. And that minimum span tree allows you to visit all the nodes in a network using the smallest steps. So this is the algorithm. So you use that minimum span tree and to move along these networks to infer the trajectory. So on your left side are the algorithms people have been developed. Okay, monocole and is the most commonly used ones. Okay. So this is the algorithm called the minimum span tree. This called is to find the subset of the edges in the connected or edge-weighted undirected graph. Then this subnet of these edges had to connect all the vertices. All the nodes had to be connected based on this minimum span tree, without any circle or with the minimum possible total edge weights. So for this particular network, the dark highlighted parse is the minimum span tree. If you add them up, those numbers, or the weights, how long this trajectory is, then you get the smallest total weight, but all the nodes have been visited, occupied by this tree. So based on this tree, you can start to build trajectories. Okay, so you covered all the subpopulation of cells using this tree, and then you select the parse. There's no algorithm can tell you and which parse you should select. You can go from 3 to to this and go different direction. Maybe there's a Y shape of a development pathways. Okay. Then there's a nearest neighbor graph based. In this case, we just look at the cells similarity, the distance. You don't even have to project them into lower dimensional space. You just compute the pairwise distance between cells. Then you just put nearest cells. So this cell has 5 nearest neighbor. You make a link between these 5 members. And that cell has a 5 nearest neighbor. You make a link. At the end, you build a huge cell by cell networks. And if this cell and that cell is too far away, there's no link. So you move all the cell population with small steps. You create this nearest neighbor network, NN graph network. And on that, you can apply minimum span tree or whatever other shortest work and to find the possible trajectories which cover most of the cells. And then, for example, you can see, okay, I want to move from one end of the network to the farthest end of another end of the network. Give me the longest path. This algorithm will go all the way, cover as much as possible cells, and build this elongated trajectory. So you can assume this trajectory is somehow capture the deviation of the cells differentiation. So for the nearest neighbor graph, if you select a k differently, see how many nearest neighbor you will count as the nearest neighbors, then you will change the change this shape. I hope I can have. See whether this works. Yes, so this example is a Java applet, and then you can change the number of the points. Okay, you start with this, and then if you join them with nearest neighbors, if you select this k, say 3, okay, 3 nearest neighbor will be considered as the nearest neighbors, will be connected. Then you start to build this network with different density. Okay, if you consider k equals 7, KNN, so 7 nearest neighbors are in the same subnetwork, then you create this much dense network compared to if you use only 2 as the nearest neighbors. Okay, is that easy? Okay, so the most important is you have to compute the distance between cells. So in this example, when k equals 3, we only consider 3 nearest neighbors, so there's a sub-isolated components in the network. Well, when you increase k equals to 4, and those nearest neighbor will find the next one, which makes this isolated component linked to the main components of the network. So that's why you can change the density of connectivity of the network of your build. So that's some parameters you want to try maybe. So this example, I use MATLAB code to build the KNN network. So if you look at those points here in each of these joint points, there are one cell, and there's a lot of cells. And then here, once they have satisfied k equals 3, then they linked. Every cell linked with the 3 nearest neighbor, and you make this network. Then I ask the algorithm to find the longest path from one end cell to the end. This is the curve found out. So it starts from this very end cells and move along this and build this trajectory for me. And after that, I can see, okay, what gene shift its expression from this end to this end. I found those genes, okay, across the cells. This gene increase, this blue gene, and they start to decrease from one end to another. And they all are NF-kappa B pathway genes, and the cells from B cells, from lymphoblastoid cell line. So this makes sense. The B cells start to, how to see, they differentiate or they active level differently, okay, driven by this NF-kappa B network genes. There are many other algorithms have been built, but in all the similar idea. I will mention RNA velocity in the next couple of slides. So this is a very different approach. They use the splicing, unsplicing rates difference to simulate the dynamic of the splicing events. Because the monocole is commonly used, I want to mention the monocole. Okay, it has been developed from 2014. I don't know, at that early stage, the authors had been already start to work on the single cell, start to build the algorithm to build. I don't know how he got this idea from, but remember 2014 has the 10x genomics technology only started from 2016, 2017. So this algorithm has been developed 3 years early than the real starting point of the single cell error. So at that stage, they use ICA, okay, to reduce the dimension and then build a minimum span tree and then to recover the pseudo time and to look at those changes of the cells. There's not many cells in here, right? But that's monocole 1. Right now, it has been monocole 3 or 4 already. But this package has never been stable. They change algorithm under the hood all the time. So version 2 will use very different algorithm. They use reverse graph embedding to do the dimensionality reduction, and then some other changes. I believe they use principal graph and principal curve to find those trajectory among cells. Monocole 2 uses a DDR tree. But anyway, let's just tell you the different version of the same package. They use completely different algorithms. So this uses PCA, the other dimensionality reduction, and then update. Select the root. So this has to be user input. You have to select the root from the graph. So your decision will be involved. Though based on you select the root, it will start to create those pseudo time from the root. So this is monocole version 2. This is monocole version 3, and the author want to make this more comprehensive, so including not only the trajectory analysis, which monocole is famous for, but also the authors want to make them into a clustering algorithm. They want to do the D analysis at the same time. So within the algorithm, start to use the UMAP and the Louvain algorithm to detect the subnetworks of the cell subpopulations. Anyway, so you need to know which algorithm, which version you are talking about, okay? Slingshot is another. I think it's time for 5 minutes break, and we come back, okay? After 5 minutes. Okay. I want to quickly show you the function that we can use through the toolbox to do the pseudo-time analysis. This will rely on monocle. Okay. The function is in here. It's called pseudo-time analysis using monocle 3 in R. You have to have that installed. So you have to set up R running environment. I don't know on this machine R has been set up or not. Then you should be able to run monocle analysis directly from using this link. There another use for the plot is you can create cell KNN network. So if I click this, say I use K equals 4, I should build the graph. This is just for visualization purpose, doesn't? Then you can based on that graph to make a trajectory, okay? It's not a clustering, it's just all the cells based on their similarity. So this is a cell-cell network. So they are not fully connected because I have so many different cell types. So if you work on a single cell type, you probably get something like this. And then you can select a trajectory. See, this is a starting point. And then the algorithm will find a path, move to this end. So fate will normally give you some trajectory-like shape of the embedding. So if you have embedding like this, you can simplify it because you, for example, you only work on the light blue cells. You can extract this cell. And then after that, you can see I can plot a cell trajectory, and this will use, for example, different algorithms to find a curve. This is too complicated for the algorithm to find all the curve. Doesn't make any sense. But if you have a trajectory and a very simple form of the cell embedding, it will get this curve correctly. And you can have a starting point, have an end point, and then you can view some genes, see which one express change along this trajectory. As I said, there's no algorithm can guarantee you get a ground truth. So any of this algorithm makes sense to you will be okay. All right? So monocle 3 could give you very terrible results. But if you use this straightforward, just look at the cells and using different embedding and build this curve in the tools, as long as it makes sense to you, that's great. Yep. Questions?
Should you pull like a population like fibroblasts and then do it?
You can if you believe there's trajectory from one end of fibroblast to another. Yeah. Yeah, so this function will take some time. Just try to find a gene, have a dramatic change from one end of cells to another. Okay, have increase, decrease. As I said, for the same problem, there are so many different ways to solve the problem no one can guarantee monocle 1 will have a better results than monocle 2. Monocle 3 maybe do a terrible job. So this all just manipulate of data points. So as long as you at the end it makes sense to you, you can use that analysis. Slingshot another way, and so this is the analysis from the examples. You found some genes follow the trajectory from that end to another, and those are the change along the time, the time response genes. Here they use principal curve. So the algorithm I showed you also have a spleen fit and a principal curve. Yeah, so you can try different algorithms. So I have no idea which one will give you the best results. Monocle 3, monocle, and been so popular. So if you want to guarantee the paper got accepted and there's no review will criticize you, maybe use monocle. But as I said, everyone come up with a different idea of how to do the embedding, how to build the minimum span tree, how to smooth these curves. It doesn't matter which one you use. Okay? Yes, Victoria.
So can you do this before you annotate?
Annotate the cell types. You can if you have, especially if you have. So I suggest to use FATE, because FATE is algorithm that help you to keep the global structure among cells. FATE. FATE is based on the MDS. It's based on the multi-dimension scaling. So this one is an algorithm to reconstruct the map. Remember, you have a pairwise distance between points, then reconstruct the map between cities. So this is an algorithm, and this algorithm is used in the FATE. So FATE will create those embeddings of the cells with this elongated trajectory. Well, the UMAP and the t-SNE will only create those clusters. This will not guarantee this this subpopulation of cells is more similar to the next one, because this is stochastic embedding. If you have, as long as the numbers in their in the high dimensional trajectory, the representation, they are close to each other, it will randomly pop up in. If you have another population cell, then there's a space, they will pop up randomly here. So if you run a principal curve or spleen fit on this, it only makes sense. So I discourage you to run t-SNE UMAP and then use this, but the fit will be better options, more justified. Yeah.
So then after you got your line, how do you look at the highly variable genes between the groups?
Yeah, so after you have this line, every cell will be forced to project itself onto this line. So each cell will have a pseudo time point. In that case, you do a regression by the gene expression along this using pseudo time, the time points as a response variables. So any genes have a strong correlation with this time will be easily picked up using lasso, for example. We can do that, yeah. So once you have the embedding of the data, then you can have the you should have this cell attributes. Because you build that curve, you should have a splitting fit pseudo time. So this is like for each cell, you have this value. So this value, and then you can see which gene correlated strongly with this value. That's the gene you want to pick out. I show you this Thursday, okay? Again, the idea is it doesn't matter, okay, which algorithm you use. The discovered biological insights are the most important one. So this algorithm claimed it can find better trajectories and make more sense using some good examples that fit their need. And T-scan, another algorithm. This is published in 2016. It's still very early in the nucleic acid research. Very clearly described the algorithm. If you follow this paper, you can easily to re-implement. That's what I did. I just look at the paper and see what algorithm they discussed, and just completely re-implement in MATLAB. Stream, another. Lisa is another. I bring it up because it's from one of my friends was in Stanford before. I don't know if it's good or not, but this is systemic comparison study, and to take all the pseudo-time analysis algorithm put together side by side, and those are the features, and those are the overall scores. So which one is give you better accuracy, whether they're scalable or not. If you have 1,000 of cells, some of this algorithm will stop running. Stability, usability, so it's huge efforts to run all these algorithms and side by side to put them and compare. This is Paga, is one of the one implemented in Python. That's the one I really don't like. But whatever. So there's continual table, okay? We go over the place. They use different metric to compare. Yeah. You see this monocle, they separate into ICA. So this is monocle 1, monocle DDR tree, and monocle 2. At that stage, there's no monocle 3 yet, but they changed again recently. At the end, they gave the guidelines. So how can you pick which algorithm you should use, making this decision tree take into account whether you expect a particular topology or not, yes or no. Do you expect the circles in the topology, or it's only just one direction, no circles, whether there's complex tree topology, yes or no. They all data scientists the guidelines, okay? So this package, which has been used systemically and put a lot of different algorithms together to do trajectory pseudo-time analysis. So it's called trajectory inference, okay? They put everything together. So this is the question. After you finish the construction of a trajectory, you have the pseudo time for each of the cells. Then what you can do next? You can look at those changes, the curves, go up, down, to look at the differential gene expression. So genes at one time end with starting point, they change dramatically. You pick those genes. You want to look at the trajectories, see whether there's, for example, the branch splitting points, see whether cells at this stage become to differentiate into two different other cell types. So look at the topologies. But remember, pseudo time is not actually time, so you probably will be looking at something different from the real time of the development. That's something we are trying to do in my lab here. So we build these cell populations, then we build nearest neighbor tree graph, then build these pseudo-time trajectories. And then along these trajectories, we subsample cells and put them into n numbers of bins. So this will be cells in early stage, then move to another time points. Then using these subsampled cell populations, we can build gene regulatory networks. And this network is from this population. So this network is from another. So then we work on the networks, time series networks, rather than the gene expression, to see whether there's some of these interactions between genes become stronger along the time or become weaker. So this is just another levels of the analysis. The way we used, including this tensor decomposition, we will mention this next week in the lecture of what the tensor is. Tensor basically is high dimensional matrix. Matrix is 2 by 2 dimension. And then if you have a 3D or 4D data set, you are dealing with a tensor structure. So you can do the decomposition for the tensor as well. So this is like a D3, then you have 3 vectors to reconstruct this tensor. To denoise, then you remove the time-related projection, then see whether you constructed, whether they are different or not. Some ideas. So this is illustration. If you look at the subtype of cells, then build the fate trajectory, then look at the pseudo-time trajectories, build the networks. These neurons probably from different layers of the brains. I want to quickly mention the velocity. So velocity is based, they claim that the trajectory inference have some limitation. Classical trajectory inference do not offer any directed dynamic information. And those trajectory inference algorithms do not take into account information beyond transparent rates and similarity. So the idea of RNA velocity is to consider this transcription dynamics. Okay, so for this particular gene, see in A, this gene is transcribed in a rate. Then you have such many of pre-mRNAs, that's before splicing. Then if you have matured mRNA in this level, and then you have degradation in the gamma B. And this gene start to be downregulated, so you start to see the decrease of the mRNA, pre-mRNA. And you have relatively more mature mRNA. Well, if you have a balanced like a steady supply of active transcription, then your spliced and unspliced version of the mRNA should be equal. So in that case, you look at this scatter plot, then you have this degradation rate and the transcription rate, and the gene which is cells in these two states are the same for this particular gene. So this gene is located in the diagonal position of here. Well, down-regulated genes will start to stop the transcription. So you'll start to see relatively few pre-mRNAs. That's unspliced mRNAs amount becomes smaller. So you have the shift of these genes dynamic towards this end of the, not in the diagonal position. Well, if you have a gene that's being upregulated, you increase transcription. The amount of the pre-mRNAs, those unspliced, will have a higher ratio compared to spliced, because you actively splice unspliced mRNAs in the pool. So in that case, you start to see increase of unspliced mRNAs compared to spliced. So based on the position of these genes in this dynamic the scatter plot to show the dynamic of the steady states, you will see whether these genes start to be downregulated or start to be increased. It's for this particular cell or cell population. Okay, if you use multiple genes abundance, you can start to look at all the genes, spliced and unspliced copies, and to look at their dynamic. So each cell may have this gene up, that gene down, and another gene in the steady states. All right, to use the information from splicing, unsplicing to infer the dynamic activity of the gene transcription. It become possible for the UMI. So UMI is only for one gene, just one rate, right? We had X matrix, right? You have genes, cell 1, cell 2, cell M. That's what our matrix is. But this matrix can be split into two, okay? So U unspliced, spliced. You have to know the eukaryote gene structure and the splicing, unsplicing if you're from data science or machine learning background. Those are the just procedures for mRNA from just transcripts and become mature. So this is a step you have to take. So this count matrix can be separated into two. Okay, the summation of these two will be the count matrix. So each gene in each cell will be separated into this copy, is spliced or unspliced. Then you look at it for the same gene, their ratio. Okay, same gene, same cell, gene 1. So for each gene, you have unspliced count and the spliced count, for example. So that's a percentage of unspliced count in each tissue. So you use that information to infer whether the cells, okay, this particular cells is in which state. So this plot is for particular genes, okay? And they look at these genes and put the cells onto this spliced, unspliced plot to look at the shape of this dynamic change. The data, because it come from different time points, and they know this genes are circadian rhythm genes, so they know their ground truth, they should be changed in different time points. So that's why they can create this curve from data from different time points. In the dark, this gene should be upregulated, so that's it shift away from the diagonal position, but upward. And in the morning, in the light, under the light, this gene will be downregulated. That's why this gene come down from these samples collected in the day, during the day. So you can see this curve change. If this gene has nothing to do with this dynamic, then it will stay on the steady states, and the cells will be located on this diagonal position. So these two examples, they are selected because they are circadian genes to show the ground truth of dynamic change. They can predict. So very quick is one of the most sophisticated, most complicated model in single cell data analysis people have built. And the algorithm has been criticized also because the implementation have to take a lot of assumptions. So see the rate of the transcription, rate of decay, how you smooth those matrices because they are so sparse, so many zeros. Move, yeah. Yes. So in order to get these two matrices, you do need extra steps. So remember in Cell Ranger will give you this directly. Cell Ranger will not give you spliced, unspliced matrix. What you have to do is to take a Cell Ranger to give you the so-called BIM file. So this BIM file is the alignment file. This is intermediate steps rather than the final output for the matrix. So normally we just throw it away. But in this case, you have to get this back because you have to count whether there's splicing or not splicing based on your alignment. In this file, you'll be using to give you these two. So it's more complicated. You shouldn't be able to do it by yourself. Okay, you need a Linux machine and to process this, to get this. But you can consider this like you want to extract the last drop of the information from your valuable data. So you have done all the routine or the analysis, including trajectory analysis, network building. You still want to get some information out, then you can see, okay, let's try RNA velocity. Unless your supervisor wants you to have RNA velocity in the paper because other people have already done it. So you pretty want to try, and it creates beautiful plots, because once you have a field, you have move, you have all the vectors, okay? You create something looks fancy, but not guarantees it's a true signal. Unless you have a good understanding of your system, especially you have a stem cell type of study, you want to look at the direction change. Okay, so this is very special. It's the immunoraptor analysis. It's single cell data analysis. If you study immunology, extremely valuable. And so here we talk about two immune cell types, and the T cell and the B cell. They all lymphocytes. They each of them, their T cell and the B cell receptor expressed on the surface of those cells. And for T cell, this should be bound with another immunofactors. Well, B cell will produce antibodies. And those receptors have been used to process information passed on by either other cells or by pathogen directly. So T cell population maintain a highly diverse population of a cell type, express different combinations of T cell receptors. And those receptor combinations have a different affinity to different kind of immunosignals. And if you have pathogen, for example, invade the system, and those information passed on, the type and amount of those pathogens will be antigens. Information will be passed on. And based on this affinity of the combinations of different receptor types, different subpopulation of T cell will be amplified, okay, specifically. So if you have bacteria, and then, for example, this T cell type A will amplified in order to gather your immune memory in order to fight another infections next time. If you have viral infections for particular virus, then you have a different subtype of T cells with different expression of the T cell receptor combinations will be amplified to specifically against viral infection. So this T cell receptor is not only one gene, it's a cluster of many genes. And these genes contain subdomains, for example, VDJ, okay? So VDJ, and there's a beta, gamma, alpha, beta, gamma, different chains, okay? So each T cell receptor have two chains, alpha or beta or gamma or delta. So each subchain contains VDJ different domain combinations. If each V subdomains contain different subtype of genes, it's a cluster, there's different slightly. So all this combined make this structure highly diverse. Okay, they all differed from each other a little bit. So the number of combinations, if you have alpha beta chain T cell receptors, then you have 7 different segmentations of V subdomain genes and 61J, and then 1 constant segmentation. Then the beta chain have 67 different type of subsections, 13 different subsections. So if you multiplicate 70 by 61 times 67 by 13, you get the potential possible combinations of those subdomains, and each have multiple copies. So the total combinatory numbers is huge. This gives you the physical or genetic background that allow you to generate larger amount of T cell receptors for specific subgroup of the T cells, each different from each other. And based on the selection of the infections of the immune response, different subpopulation of T cell will be amplified, expanded this colony greatly. So this is a genome browser view of the human T cell receptor gene cluster, just a very small part of this. And you see the T cell receptor B, okay, beta chain V6-4. And then you have a T cell receptor beta chain V segmentation 7-3. So there are many of them, okay? T cell will selected express some of them, make combinations of T cell structure, receptor structure. Similarly, so this is for alpha chain, and this is for gamma, okay? So this is illustration of those combinations from clusters of this receptor sub-segmentation. Then you have different combinations at the end. This is just one of the subtype of a T cell receptor for this particular T cell population. And there are different ways to make another. So if you count the possibilities, then you have, for alpha chain, you have 10 to the power 7 combinations. And here we have beta chain, you have 10 to the power 14 combinations. Altogether, you have such a large amount of potential to differentiate each individual T cell, their receptor. So we want to, at a single cell level, if you have a T cell, you want to know what's T cell gene expression. This is single cell RNA-seq. You also want to know what surface protein, the T cell receptor, which subcolony, what's the combination of those. So that's why 10x Genomics have these solutions to allow you to do this T cell receptor or B cell receptor amplification kit, specifically target T cell and B cell. So you get the population, you get the X matrix from T cells. At the same time, for each T cells, you get its receptor subdomain combinations. So which type is this? The idea is, if your T cell is in the naive form, so this gene expression, in the same time for each cells, each T cell, for example, it will give you the label. This so you have a TR, so alpha beta 70. I just made it up, and this is a TR alpha beta 24, 25, whatever. So each will be different from each other. So this population has no, it's naive or standard position. But if you have a population which has been challenged with antigens, either it's the cell population is fighting with a particular specific type of cancer, or it has been just selected for against particular antigen infections, you will see quite a lot. See, 50% of all the T cells have this combination type, T cell receptor alpha beta. See, 27, 8, 6. So I just made it up. This is unusual. That tells you this particular T cell population get unproportionately expanded colony. So in your sample, supposed to have a different cell type, T cell receptor combinations, but now the majority of them is this particular given type. That tells you this particular colony has been unproportionately amplified or expanded by the challenge of the problem you studied. So this combination will give you some clue that that T cell receptor particular combination will be very informative against that particular antigen. Okay? So you want to know what type of T cell will fight the cancer cell most effectively using this method. Same thing for the B cell. B cell also have the B cell receptors. Okay? Just named differently. So this is alpha, beta combinations of this. So we want to get information from there. So how can we do that? So this is a particular protocol we have to follow in order to get both expression matrix and also the subtype of a T cell receptor. You have to have extra steps after a single cell RNA-seq. So this is the same as a single cell RNA-seq. You label, use barcode, and then the same thing, okay? You create this master mixture labeled with all the 3' poly-A's. Same procedure. And the amplification and to build your GM, the droplets, same thing, okay? But now you have, so they're all the same. Here you generate okay. So next step is different. So you prepared your droplet already. Now you separate it into two tubes. One will go to regular single cell RNA-seq. You get the expression matrix. That's what we did. Another tube you will go, and you know your sample is all from immune cells, mostly T cell, B cells. Next, the separate tube will go to amplification for the receptor genes. Okay, you separate your master piece of the solution into them. Let's see whether you can get exactly the same T cells. You will get exactly the same T cells. Because the same T cells has been, one T cell has been labeled with the gel bead barcode oligo. And all the mRNAs will be labeled with this barcode. And the solution will be mixed together, and you equally separated this solution into two tubes. So the transcripts from the same T cell will be separated equally in theory. Each contains half of the information of the expression itself. So for the same T cell, each cell will labeled with the same barcode. See, A. So this will be ACTTA. This will be CGAAT, whatever. But this, all the contents in there will be separated into two. Then for this tube solution, you have to do another round of amplification, specifically designed the primer for the VDJ, okay, region. So this will enhance to do another levels of the amplification, specifically increase mRNAs from that given region of that particular gene. this gene has been labeled with the cell barcode. So at the end, you still know what this gene is from. Yeah. Yeah, I think in the standard preparation at this step, you break up the cells. Everything will mix. All the differences of these mRNAs have been labeled. Each is labeled with this barcode. So at the end, you will be able to tell whether those copies of mRNAs are from the same cell or not. So these copies, some of them from T cell receptor genes. So you just use another tube to specifically amplify those transcripts in order to read out the different segmentation types. Okay. So this experiment will be double the charge, very expensive. You basically use one sample, you did it twice, two different single-cell RNA-seq experiment. But it's valuable for people study T cell, B cells, right?
So is the enrichment level you use the kit? What do you use the kit?
The kit is this specifically in amplified receptor genes.
And when?
After you prepare the solution, after you label the cells, and for the single cell RNA-seq is ready to sequence almost. On this side, you do one more step to.
Yeah, that's one. It's separated tubes.
Yeah.
This kit is cell tubes, then you use the kit, right?
Yeah, to use this kit to amplify the gene. Okay, so this is a principle. At the end, you will use a cell ranger, and the cell ranger have a special version for cell ranger VDJ. So this will read these labels and put the cells, and also read out what type of these combinations of receptor subdomains. Those are the example files we'll generate from the VDJ. At the end, this will be one extra table. So you get the X matrix expression. You will also get one of these Excel spreadsheet. Okay, you spend double the resource money, and you get one spreadsheet. This spreadsheet contains each cell's barcode, okay, and then tell you whether it's a T cell or then what alpha chain type, what alpha the beta chain type, what are the combinations, what clonal type it has. So within the transcripts, you will see those clonal type reconstructed. If you search this, you will find in the human or mouse genome where this particular alpha chain, beta chain, which type it is. Yeah, so I think this is example results. All the cells, these T cells, from one subclone. So this is extremely expanded, this particular colony. And you know the type. So this is an example. So this combinations of the type, the frequency, this may help you to build better vaccines. Maybe you know the surface protein. So this is another view. So those are the same colony of T cells put together. So you know this reaction amplified this subtype of T cell, this subtype of colony, and then there's some other cell types. Not the cell types, different T cell receptor colonies. Okay, that's all. Yeah, you have the same thing for the B cell as well. Okay, B cell is another, the same. So you triplicate your cost in order to get that information. All right, thank you.